here with Frank Patelka up to 1988, and then continued on as a postdoc with Patelka and Monty through to 91. He went on to San Francisco State and now is at UCLA, where he's the acting director of the Institute of Environment and also director of the Center for Tropical Forest Research, and a new one on climate change solutions. Some of his contributions have been really broad and I think fascinating and influential. One on ecological genetics of finches. He's done a lot of work on evolution in tropical forest mosaics, and that's led him to his interest in evolution in human-altered environments. And that's what he's going to tell us about today. Welcome back, Tom.
Thanks, Greg. It's great to be back, and I have to say it's a little intimidating to see so many members of my thesis committee here. I hope you don't follow up with any of the questions that you asked during my orals. But it's wonderful to be back. It's estimated that about 2/3 of the world's terrestrial land area is now devoted directly to supporting human populations, either through urbanization, infrastructure, or agriculture. And as a consequence of these impacts, we're witnessing a global but unplanned evolutionary experiment with the biotic diversity of the planet. And it's pretty clear, I'm sure most of the folks in this room, that these impacts are impacting every corner of the globe. Classical examples of rapid evolution driven by humans, such as industrial melanism, once considered atypical, are now becoming commonplace. We're seeing size-selected fishing affecting the genetic basis of growth potential in exploited fishes. Noise pollution is resulting in vocalization differences between forest and urban dwelling birds. Recent research also suggests that human disturbance may have a negative impact on biodiversity by homogenizing the environment, inhibiting the process of diversification. There's many recent examples of this. Andrew Hendry's work with the grants on Geospiza fortis on the island of Santa Cruz in the Galapagos showed that over time the population of fortis has lost its bimodality because of human population growth there. In Lake Victoria, Ole Schuhausen, working on cichlids, showed that eutrophication of the lake and the lack of visibility has resulted in a breakdown in assortative mating. And this is also the case in cichlids I should say sticklebacks in North America, some work by Eric Taylor, who showed that there was a breakdown because of visibility in these lakes due to introductions of crayfish between benthic and limnetic forms of stickleback. The objectives of my talk today are really to do several things. I want to examine evidence for evolutionary change in human-altered habitats and its consequences. And I'm going to give you sort of a grand tour of some of the things my lab is involved in looking at this question. First, we're going to examine brown anoles in the Caribbean, move on and talk about a rainforest bird in Central Africa, and then talk about how humans may be altering ecological gradients overall in both, I'm going to give you some examples from Africa and South America. And then finally, I'm going to wrap up talking a bit about ways that we might incorporate evolutionary process into conservation planning and some new frameworks that we've been developing down at UCLA. First of all, I'd just like to mention many of the collaborators, especially on the Africa work and the gradient work, many people working in the field, in the lab, and also doing various kinds of modeling. For the lizard work, a lot of the work I'm going to be talking today was done by Minooka. She's a graduate student who just finished her PhD, and also collaborative work with Ryan Kalsbeek and Camille Bonneau, two postdocs who have since moved on but worked on the lizard project. Well, let's talk about impacts on islands to begin with. Obviously, islands, more than any other ecosystem, have been severely impacted by humans. What we've been focused on is to use the anole radiation to begin to look at this. Of course, anoles show a magnificent adaptive radiation over 150 species, some many morphotypes that are adapted for different types of habitats. What we chose to focus on is the brown anole. This is Anolis segrii, and what we've done is to look at patterns of selection and divergence in this lizard, and also to see if we can discern genetic differentiation as well, and levels of plasticity. This shows some of the study sites that we've been working on. These are 4 islands in the Caribbean, and we've been comparing disturbed habitats with natural habitats. So disturbed habitats runs the gamut from you know, a boat dock to a park behind a hotel, so various types of disturbed habitats. And then natural habitats would be characterized by the natural vegetation with trees of various diameter. Some of our sites are pretty bizarre, as you can imagine. You know, this is, you know, actually we didn't work here, we worked at the Club Med next to this place. But it is amazing. You can go to these sites in the Caribbean and, you know, completely artificially constructed, and you can find Anolis populations doing just fine running around on the rocks. You can also, if you get tired, sit down and have a margarita if you get some coffee. If we look at the islands, just differences in snout vent length and hind limb, we find significant differences between disturbed and natural sites in overall body size, and when we control for body size in hind limb as well. So we're getting these dramatic morphological differences in these different habitats. Now you might say, well, maybe this has to do with simply colonization. There might be differential colonization of different populations. But in fact, when you look at allele sharing distances based on microsatellites, all the different populations are derived from the same population. And so these processes that are changing morphology are clearly happening in situ. To get a better sense of how selection may be acting in these 2 habitats, we focused our attention on great exuma and had 5 sites, 3 disturbed sites and 2 natural sites that were separated by about 2 kilometers. And we marked individuals using elastomeric tags. These are a dye you can inject under the skin, and under a black light you can identify individuals this way. And if we look at selection coefficients for linear and nonlinear forms of selection, we find a very contrasting pattern between natural and disturbed sites. And in this case, in natural sites, we didn't discern any pattern of selection, but in disturbed sites we had 1 instance of disruptive selection and 2 instances of directional selection. Now in these comparisons, the coefficients were calculated from separate regressions of survival against hind limb with snout vent length as a covariate to account for body size. We can look at this in a little bit more detail. If we look at the phenotypic distributions and fitness surfaces and perch diameters, you can see natural habitats here with the total histogram, all lizards captured, and then the white histograms, those that survived. And you can see patterns of directional selection, for example, using the spline here and hind limb. And also you can look at how the survivors of these populations, how they're using their perches. And so you can see that the survivors are indeed using, in this case, smaller diameter perches. Well, we were also interested to see if we might be picking up any kind of microgeographic pattern in genetic differentiation. And we were really, quite frankly, surprised to find that we picked up positive, significant FSTs when we compared natural and disturbed sites. In all cases, and in 1 case, we even picked up differences between disturbed sites. If we do the obligatory structure analysis with a k equal 2, you can clearly see that something's going on differently in natural sites than in disturbed sites. Well, the other question we had, of course, was to what extent are these morphological shifts just having to do with differential plasticity in different environments? We know, for example, that that the human disturbed sites are much warmer, about 3 degrees C warmer than natural sites. So this might be a contribution. There might be food differences. We were also interested in also seeing how heritable hind limb was. And so what we did was to bring lizards in from Great Exuma into the lab and conduct breeding experiments, and with an experimental design looking at changes and looking at growth rates at different temperatures and food regimes. So I think we started with about 40 or 50 males and 40 or 50 females for this breeding study that went on for a couple of years. Well, heritabilities, first of all, were significant, so obviously hind limb can evolve by selection. And evidence for plasticity, this was basically the design of 196 days. We looked at combinations of high and low food and high and low temperature. And what we found was really quite interesting, and actually interesting in the sense that we found dramatic differences in size dimorphism. So in general, what we found was that in females, there was very little variation across these different experimental designs, whereas in males, there was this tremendous difference. So you got much larger males in a situation with high food and high temperature. But just the extent of range of sexual size dimorphism here is quite dramatic when you think about how food and temperature might be affecting this. Now, if we look at hind limb, the character of particular interest and control for body size, this plasticity goes away, and we don't find evidence of plasticity in hind limb. So, of course, this is just dealing with food and temperature. And, but we know previous work by Jonathan Losos in a paper in Evolution in 2000 did show that there was a differential growth rate in both males and females on broad and narrow surfaces. And so I think we still have a ways to go in understanding the role of plasticity in this system. But just to summarize some of the Anolis work, it's pretty clear that human-driven habitat changes have impacted both the strength and form of natural selection acting on hind limb. In contrast to previous work, human-altered habitats can produce diversifying selection, and we see this in one instance. Also, we're finding fine-scale genetic differentiation between these populations. Still, we need more investigations to really understand the context of that. And then traits under selection have a clear heritability and evolve, and more work needs to be done on plasticity. And particularly, what we're hoping to focus on now is looking at individual genes under selection. Okay, so that's the lizard example. Now I'd like to switch gears and talk about another example, another project where we've been looking at evolution in human-altered habitats, and this has been work that's been centered in Central Africa, and this really examines this whole effect of fragmentation that's so, of course, rampant worldwide. If we look at just tropical rainforests in general, we find that 50% of the world's rainforests are in various stages of pasture abandonment or secondary forest growth. And you know, we tend to think about fragmentation leading to local extinction of populations, but for the species that persist in these habitats, clearly we're imposing selection in various ways, and so we may be changing the evolutionary trajectory of these species that do persist in these degraded habitats, and that's what we wanted to look at. The species that we've focused on is one we've been working on for a number of years, is the little green bull. It's a species of bulbul. It's a medium-sized bird. It's common in both secondary mature forest, seen here with this open understory, and secondary forest. This particular one is a cacao plantation with an open canopy. This is an understory bird, so it's foraging down here on fruits and insects. If we look at our study sites, first of all, we wanted to define mature and secondary forest, and we did so with remote sensing variables. So for example, tree cover, for MODIS data for tree cover shows mature sites at about 80% canopy cover, whereas secondary sites are about 65. The fairy index, also a big difference. And so this is how we define our mature and secondary sites. And you can see this is a MODIS tree cover map of Central Africa, and it's not great resolution, but I think you can see that what you find here in this rainforest region is really very much a mosaic between secondary and mature forest. If we look at abundance first in these two different habitats, we find that bulbuls are about 3 times more abundant in secondary forest than in mature forest. This was interesting, certainly something to follow up on. We know there are many examples of density-dependent selection that people have been able to show in wild populations. So we're finding differences in abundance that might be of interest. We're also finding, we also found many differences in morphology. Wing, tarsus, and bill were significantly different. Also significant differences in size and shape. We don't know yet what these differences are related to. For example, wing length, larger wings and a more open understory makes sense in the sense of aerodynamic efficiency. Birds in more open habitats will have longer wings, but just more work that we need to do to understand the patterns here. We did want to take a closer look at what some of the environmental, some of the remote sensing variables, how much of the variation in morphology they could explain, and we used generalized similarity modeling to look at this, and it turns out that up to 60% of the variation in bill size, for example, can be explained by environmental variables. Again, we don't know the specifics of how those variables are interacting with morphology, but there is a clear signal here that environment matters. We looked at song duration. We were interested in differences in song and found significant differences in duration between mature and secondary habitats in both of the major song types. Birds tend to sing a longer song in mature sites than in secondary sites. This could be due to the fact that in secondary sites you have an open canopy with more wind turbulence, so it may well be that we know from work on sparrows, for example, that wind turbulence in more open sites causes a shorter duration song, so there may be effects like this going on at this microgeographic scale. We also wanted to look at coloration to see how this might be changing. This is collaborative work with Greg Brother, who knows a lot more about color than I do, and we looked at tail feathers as well as wing coverts and found that chroma was significantly different between secondary and mature sites, with higher chroma in secondary sites than in mature sites. Furthermore, based on Endler's criteria, these differences are of a magnitude that the bird should be able to recognize. And so slight differences between the sexes, no real big differences there. We had some differences in luminance. It turns out greater variance in luminance than in secondary than in mature sites. These are still things we're following up on. Like the lizards, we're also interested to see if we could find microgeographic differences in structure. And so we used a whole genome scan using AFLPs, looked at 84 individuals and 63 loci. And we do get separation between secondary and mature forest. Looking at the structure results, again, we find that the orange area in secondary forest, the green area in mature forest, we are finding with a K=2 that we can distinguish these populations. We were also interested to see if perhaps some of the genetic variation could be explained simply by isolation by distance. So we did a distance-based redundancy analysis. This is a multivariate technique that Anderson developed to look at environmental factors on genetic differentiation. And so when we tease out geographic distance, we still find that environmental variables are significant in most cases. We also looked at evidence that particular loci might be under selection. Using the Beaumont and Balding technique, we found, this is an outlier analysis, we found that two loci, locus 53 and 42, appear to be under selection or linked to genes that are under selection. Of course, we don't know what traits these might relate to. This is going to take a lot more work, but nevertheless, it does look like we're finding differential selection at the genomic level, which I think is quite interesting. Finally, we're also interested in disease differences. We have a big project looking at various diseases, and it turns out with Andropodus virens, the bulbul, they have rather low parasitemias, and so we decided to look at another of our target species. This is the olive sunbird, Siamitra olivacea, and look at two blood parasites, Hemoproteus and Leucocytozoan, two protozoans. And Hemoproteus is transmitted by biting midges, and Leucocytozoa by simuliid flies. And what we found was that the prevalence of both parasites was much higher in mature than in secondary sites. Preliminary data that it wasn't ready for today's talk also shows that there are certain haplotypes that are more frequent in secondary than mature sites. So I think this is quite interesting. We want to follow up on this. Why do you have these differences? We're not sure yet. It may well be a breakdown of host-vector-parasite relationships. When you move into secondary forest, you have a lot of human-adapted vectors that may outcompete perhaps the ones that you find in mature forest. We're not sure, but anyway, this is a dramatic pattern. This was based on 10 paired sites where the sites were separated by about 5 kilometers, 5 to 10 kilometers. So to sum up green bulls, habitat differences between mature and human-altered secondary forests have resulted in shifts in abundance, morphological traits, vocalizations, plumage coloration, genetic variation, in the case of sunbirds, disease prevalence. Okay, well now I'd like to turn to the last portion of the data part of this talk, which has to do with environmental gradients. We're all aware of the fact that natural selection along gradients can be a powerful driver of adaptive variation and in some cases speciation. And clearly because of given climate change, preserving them may offer a bed hedging approach, the idea that perhaps some populations along a gradient are going to be adapted to the new climate, whatever it is. So we've been working on gradients for many years, and only recently have we decided to look at the anthropogenic impacts on gradients to see how they might measure up. The first gradient I'm going to talk about is one that we find in Africa. This is a gradient between rainforest and ecotone habitat, or savanna habitat. Ecotones represent a vast area of transitional habitat, mosaic habitat, about 18,000 square kilometers that form a kind of ecological sandwich around the Congo Basin. And there's also pretty good evidence from Jan Feltz's work, early work, showing how avian diversification and new species are often associated with this particular gradient, and also gradients associated with elevational gradients. So there's evidence that these gradients might be very important in diversification. And what I'd like to do is just fill you in a little bit about some of the past work we've done on gradients and then show you how they might be changing. This shows you some of our sampling sites in Upper and Lower Guinea. I'm going to be talking about primarily Upper and Lower Guinea. I'm going to actually, I often refer to Upper Guinea as West Africa, and this is Central Africa, but you get the idea. This is the kind of habitat you find the virens living in the lowland rainforest. Some of the sites that we visited are really pretty amazing. You can actually mist net along elephant trails in some of these areas. It's really stimulating. This is a typical ecotone site. This is a forest fragment that's floating out here in the Sea of Savannah. Yet if you were to go into this fragment, you may find many of the same rainforest species that you'd find in lowland forests, but they've been sequestered in these fragments that are associated with riparian areas and so forth. And obviously, selection, you might assume, might operate very differently in a habitat like this than in a lowland rainforest. If we look at divergence, morphological divergence as a function of distance, in the little green bowl you find that forest ecotone populations are much more divergent per unit distance. So even populations that are separated by over 600 kilometers, forest-forest comparisons are less divergent than ecotone forest populations, comparisons that are 100 or 150 kilometers apart. We also found good evidence for the divergence with gene flow, at least a component of the divergence with gene flow model of speciation in this species, where we find that ecotone rainforest comparisons are more divergent per unit gene flow than either within habitat comparisons, ecotone, or rainforest. You also find this homogenizing effect of morphological divergence of gene flow on morphological divergence as gene flow increases. Recent work on using AFLPs to look at sort of whole genome scans rather than 10 microsatellites. We're starting to pick up a differentiation between forest and ecotone populations. So clearly, we're seeing not only morphological divergence, but we're also picking up genetic divergence between these populations. Now, obviously, divergence doesn't necessarily lead to speciation. It may not, but we were interested in evidence for reproductive isolation between these, and so we looked at breeding activity in ecotone versus the forest, and what you find is that the breeding activity actually flip-flops the breeding season, and that has to do simply with an asymmetry, the fact that rainfall is flip-flops as you move from the ecotone to the forest, and so birds breed when it rains. The rainfall is at different times of year, and as a consequence, you get differences in timing of reproduction. We've also found differences in acoustic divergence between rainforest and ecotone habitats. This is work that was spearheaded by Hans Slaberkorn, now at Leiden University, where he found differences in maximum frequency, minimum frequency, delivery rate between the two major song types. So birds are singing a very different song between these two habitat types. Now at first, we thought, well, this just has to do with the structure of the vegetation. So what Hans did was to construct an artificial song and do playbacks through rainforest and ecotone habitat at both low and high frequency, no differences. And so there doesn't appear to be an effect of the habitat itself, the physical habitat, on the amplitude and frequency of what's going on with these birds. And so what we discovered, however, is that there's a very great difference in ambient noise between these habitats. And that's because when you're in the rainforest, you get a lot of these guys, cicadas, and other insects that just obliterate certain frequency ranges. And so what we believe now is happening, the reason for these frequency shifts, we believe, has to do with ambient noise. And obviously, if you're a Viren singing at 5 kilohertz, you're going to have a hard time getting a mate. And so we think that this is the ambient noise is what's driving differentiation. We've also recently done some playback experiments. These are pretty interesting, where we played back the song of Cameroon forest birds and Uganda, or I should say Cameroon ecotone birds and Uganda forest birds to Cameroon forest birds. Okay, so that's what it is. We're working on forest birds in Cameroon. If you play back a Cameroon forest bird to a Cameroon forest bird, it has a very strong response. It recognizes it as a congener and attacks the speaker. Even if you play back a Uganda forest song, and this is, these Uganda birds were recorded a few thousand kilometers away, you also find a very strong response. And so, birds from the same habitat basically are responding irregardless of how distant they are. But if you change habitats here and drive 50 kilometers north into the Ecotone and record a green bull song and play it back to a forest bird, there's very little response, or much less response. And so clearly we're seeing that these birds are differentially recognizing individuals based on habitat. So in terms of evidence for reproductive divergence in the green bull populations, and this is based on Coyne and Orr's criteria, clearly we have prezygotic isolating variants having to do with habitat. We have temporal patterns, the differences that have to do with the timing of reproduction, and we've also shown that there are behavioral differences between forest and ecotone populations. Just want to mention a few other species. We're working on a whole suite of species, but we're finding similar patterns. This is data from the olive sunbird. Again, greater morphological divergence in forest ecotone populations than in forest. And then this one's for Ted Papamphus. Oop, not this one. This is, well, maybe you like sunbirds. I don't know, Ted, I doubt it. This is also the Maxent prediction of Plasmodia hemocrotius in olive sunbirds. And the only thing I want to point out here is that this shows the very distinct gradient as you move from the forest into the ecotone. So again, on this larger scale, we're finding differences in prevalence of avian malaria. This model, by the way, turns out was extremely accurate for predicting the probability of malaria in olive sunbirds. We sampled in Cameroon, sampled in Ivory Coast, and then we recently sampled in Ghana. And you know, the model was dead on. We could go to a forest, a section of forest, and basically predict what the prevalence would be based on satellite and bioclimatic models. So this one's for Ted. This is the Mabuya slide, which is now called Tricholepis, and this is work that one of my graduate students, Adam Friedman, has been working on. And again, we're finding divergence along this ecotone forest gradient. Here he actually breaks it up into lower and upper ecotone, and also his recent work also shows that there are genetic differences between rainforest and ecotone populations. So how might these gradients be affected by human activities? Clearly, we're seeing a great deal of deforestation in tropical rainforests. About 1.5 million hectares are lost every year. And so the question is, you know, this very important gradient that's producing adaptive variation, is it flattening? Is it flattening as a result of these perturbations? And also, what are the evolutionary consequences of this flattening, and can we measure it? Well, if we look at changes in NDVI, which, by the way, NDVI is a measure of greenness, but correlates very strongly with tree cover, what we find is that the major areas of loss of rainforest are in Ivory Coast, in this area, in West Africa, some in Cameroon, but nowhere near as extreme. And so we wanted to look at this and say, well, you know, from this we should predict that the gradient should be much flatter in this region than in Central Africa. And so that's what we did, was to try to contrast these. The causation of this flattening in Ivory Coast, if you've ever been to Ivory Coast, it's you know, a large palm plantation. I mean, oil palm has been has just obliterated much of the forest region in this area, much less so in Central Africa. And we can also estimate the extent of rainforest historically. This is using some climate modeling work that shows the extent of rainforest, and this approximates what rainforests look like in historical times in the green here. And then we can contrast that with MODIS data to look at tree cover. And what you can see is, you know, most of the rainforest area in Ivory Coast has been lost. So there's been a tremendous amount of forest loss. So what are the consequences of these sorts of changes? Well, if we look at morphological diversity in olive sunbirds and in Andropodus virens, for Central Africa, compare that to West Africa, we see very clearly this flattening. In the West, much fewer, much less of a change between forest and ecotone habitats, where in Central Africa you still get this dramatic shift in morphology between forest and ecotone. This is, I can show you this a little better in the next slide. If my computer didn't crash here. Okay. This shows you the difference between ecotone and forest populations in West and Central Africa. So the black bars are Central Africa, and you can see still significant differences, or still large differences, but significantly different from what we find in West Africa, where differences between forest and ecotone populations are much diminished. Okay, so that's the pattern in Africa. It looks like we're getting gradient flattening. We're losing adaptive diversity along these gradients. What's going on in South America? Well, again, Jan Feltz's work, his early work looking at intraspecific diversity in birds, identified the Andes and the gradients associated with the Andes as very important areas of diversification. And we've been working on this question with a number of colleagues. And what I'd like to do is talk about the gradient, contrast the gradient in eastern Ecuador with western Ecuador. So what's happened in western Ecuador is that there's only about 5% of the forest left in this area. Whereas in the east, it's largely intact. There's still obviously habitat destruction, but what we're going to be doing in the next few minutes is just contrasting eastern Ecuador from western Ecuador and talking about what's going on with morphological and genetic divergence. The species one of the target species we've been working on is the wedge-billed woodcreeper. This is a little bird that feeds on the trunks of trees. We looked at 196 individuals at 29 sites. This is work that was spearheaded by one of my graduate students, Borja Mila. And these birds live in both lowland and foothill habitats. So lowland up to about 500 meters, and then foothills up to 1,500 meters. If you look at morphological shifts in this species, what you find is significant shifts in both tarsus length and bill length, with longer-billed, longer-legged individuals in foothills. This makes sense given the habitat that these birds reside in. In the foothills, you find a great deal of moss on the trees, and so we think this shift in morphology has to do with performance on these different substrates. In the lowlands, there's most of the trees are moss-free, so longer legs, longer bills in the foothills. Now, what we've done is again to use this GDM analysis to project morphological variation and genetic variation across these different habitats on the eastern and western side of the Andes. And this is a very useful tool because it gives you an idea of what morphology and genetic divergence may be doing in places where you haven't sampled. I know folks here at the MDC have been using GDM, but you can develop a predictive landscape for some of the characters by combining it with environmental variables that we get from remote sensing data and bioclimatic data, such as various forms of satellite percent tree cover, backscatter, shuttle data, etc. Well, when you do this and use a GDM model, this is the pattern in eastern Ecuador. So we're looking to the west. This is eastern Ecuador. This is the Andes up here. These are showing some of our sampling sites. And what we find in this undisturbed gradient, we're finding about that environmental variables are explaining 70% of the variation in tarsus, with most of the change occurring along this gradient. And the way to interpret these maps is that as you go from green to blue, basically you're going from a small tarsus to a long tarsus. And so you can see that the most rapid change, the greatest turnover in tarsus, is occurring along this gradient. And you can do the same thing for genetic data. This is some AFLP data. and again explains a great deal of the variation in AFLP variations. Sort of, we're still looking into this to make sure this is correct, but nevertheless, what we do find is, again, evidence of this strong gradient between the lowlands and the foothills. We also find this sort of north-south pattern too, but the most abrupt change is occurring along from the lowlands up into the foothills in terms of genetic differentiation. So the question then is, what's happening on the western side of the Andes, where you've had a great deal of disturbance? And what I've done here is just to show you these GDM landscapes for the east in undisturbed tail and undisturbed tarsus. You can see that there's a very distinct gradient. But what happens in the west, where there is a lot of disturbance, you get this homogenization effect, where you have certain areas where there's really no difference between the lowlands and the foothills. You have this lighter blue, which is not showing much differentiation, and then you have this kind of bizarre mosaic effect of high and low diversification that probably has to do with patterns of fragmentation and other things going on. So that's what we find with morphology. When we do the same for the AFLP data, again we find this homogenization between lowlands and the foothills. Very, very different from pristine areas. So in sum here, I think there's pretty good evidence to suggest that these major these major ecological gradients are flattening, and we're seeing a homogenization of phenotypic and genetic variation, and I think in many cases a reversing of diversification. Further losses of variation may also limit the ability of these populations to adapt to changing climate. I mean, we're losing the evolutionary potential to adapt to a new climate as we homogenize these gradients. So I'd like to just finish up and talk a little bit about what we can do about this, or how we can begin to perhaps incorporate evolutionary process into conservation planning. Obviously, we have to ask what the goal of conservation should be. Is it to maximize a given species' abundance regardless of the evolutionary consequences? If this is the case, I mean, this is the way, unfortunately, a lot of conservation organizations are functioning right now. If this is the case, obviously, for Andropotis virens, secondary forest is great because they're 3 times more abundant. Or should we be trying to maintain populations in a more pristine state in which evolutionary processes act in accordance? Or do we need to sort of find some common ground between these two scenarios? In terms of obstacles for incorporating evolution into conservation planning, I think there are obviously very different time frames. Policymakers and conservation planners think in terms of years or decades, whereas evolution typically proceeds, of course, more slowly. Also, conservation actions tend to be directed toward the status quo rather than adopting a more dynamic approach that would encourage diversity and change and be more consistent with an evolutionary perspective. If we want to put evolution into a conservation context, obviously if we try to put evolutionary process at the heart of conservation planning, and maybe we need to move to a more dynamic mode of environmental management. I mean, perhaps, some people have suggested that we need evolutionary impact statements before a major project is undertaken. Realistically, I think we're a ways away before we get to incorporating evolution into conservation planning to that extent, but the hope is that we can work around the edges and begin to incorporate evolutionary considerations into management plans. And I always have to, I love to quote my good friend Mark Reynolds, who was also an alum of the MVZ, who works for the Nature Conservancy, who always likes to tell me that if you can map it, you can conserve it. And so that's what we've been trying to do recently, is to try to come up with a way of mapping evolutionary process that can give conservation decision makers some some tools that they can use in preserving areas. And I just want to briefly mention some of that, how we're doing this. This is a collaborative study looking at birds, bats, frogs, small mammals. We're using some distributional data from primates and trees, focused on Ecuador. And so we're combining point locality data, genetic and phenotypic data, along with bioclimatic data and remote sensing data to see if we can begin to identify areas important in the process. This is the framework we've come up with. It looks complicated. It's actually quite simple. It's over here in this tan area is basically the hotspot approach that everyone's familiar with. You look at species richness, levels of endemism, assess degree of threat and opportunity, and come up with a final prioritization scheme. This box represents how we're trying to incorporate evolutionary process into this. And there are two elements to this. The first is to basically come up with a really good distribution model for species using algorithms that are widely used, such as Maxent, using environmental variables, and then model it using this GDM approach, this nonlinear regression technique that I've mentioned, to then combine information on the pattern of species distributions along with what we can understand about genetic diversity and phenotypic diversity and integrate this into a more substantive approach. Well, this shows you just a Maxent distribution for the wedge-billed woodcreeper, showing the probability of occurrence in Ecuador. And you know, if you have good point locality data, you know, if you're standing there netting a bird and have a GPS, it's amazing the kind of resolution you can get and how accurate these are. I mean, when we compare these to the very best distribution maps from ornithologists, we find that they're dead on. And they also often indicate areas where ornithologists have missed populations. But just to be on the conservative side, we take a 10% mask for the distribution. We don't want to be estimating process in a place where the bird might not occur. And then we use this GDM modeling, as I've shown you already, for, in this case, wedge-billed woodcreepers, for tarsus and genetic variation. And then we can combine this with a bunch of other species, such as a masked flower piercer, a species of diglossa, where we use microsatellite data and morphological data to again map this turnover in variation. And here's another map that we've developed from street neck flycatchers using microsats and morphological variation. And when we put all of this together, we actually discover some pretty interesting things. Now, we're only looking at 3 birds right now. We haven't added all the frogs and other things that we want to add. But what we can identify already, just with just a few species, is that we're missing areas that seem to be very important in adaptive variation and high turnover. These stippled areas are the protected areas, and what we're finding is in Ecuador is that areas in northwestern and northeastern slope of the Andes look to be really important in terms of turnover in morphology and genetic variation. Now, the other thing, just parenthetically here on these models, is that they also lend themselves to understanding patterns of climate change, because you can input you know, a different climate scenario into this. So, for example, this is the predicted range of the speckled hummingbird based on 150 sites, showing its distribution. And you know, just using some of the models that are available, this isn't very sophisticated. But if you look at what variables matter in mapping this species, you see that mean annual temperature is very important. Minimum temperature of the coldest month is important, and so forth. And you can then develop new predictive maps based on what the distribution of that species should be with a doubling of CO2 in this case. So here, for the speckled hummingbird, we lose 12,000 square kilometers of its native range. But you know, in any event, this is the kind of thing that you can incorporate into this framework that we're going to be working on. And now, currently, we're focused on how we can actually model process under different climate scenarios. Finally, where are we going with all of this? I think the really exciting thing for us now is to really get down to the gene level and look at adaptive genes across these landscapes. We recently went together with some other folks and bought a 454 sequencer, and we're sequencing away, spending gobs of money, and we're excited about using these burgeoning new technologies to understand pattern and process even further. So I think the time is late. I think I'll end there and thank everybody for coming.
Thank you, Tom. That was terrific. Some folks, the including me, have to go to classes. You lead, and then Rory, I think, will take over and moderate questions. So thank you again, Tom.
Yeah.
Tom, you make the point about human
activity. Is this an
activity.
orals question? I just have to.
What?
Is this an orals question?
No. For me, yeah. You make the point about human activities tending to homogenize the gene pools of differentiated areas, but it doesn't follow from that necessarily that you get a loss of genetic variation that you could be used in future evolutionary adaptation, does it? I mean, it seems like you increase the variation.
Yeah, I mean, this is the conundrum here, I think, that we're facing. In a certain sense, if you look at along the gradient, I think you are overall homogenizing things, and we can show that. But under a fragmentation scenario, what you may be doing is actually increasing variation, as I can show in Andropodous virens. And so, what does that mean for conservation decision makers? And is it? I mean, there's no question that climate change is going to turn evolution on its head. But I think we have to get out in front and try to understand, what we're going to do in terms of maximizing adaptive variation. And I would hope that we didn't maximize it by turning primary forest into secondary forest. But in fact, it does lead to greater diversification in that case. So it's a good question, and it's one I've been kind of troubled with in terms of what we want to do. Yeah.
Obviously, the human disturbance will have a huge impact, demographic impact on the natural population.
Right.
And I was wondering, because for a bottleneck, for instance, you can limit a lot of different type of selections. So I was wondering how, can you explain what you call adaptive, we call adaptation? Can you explain them just
through some bottlenecking?
Yeah. So
yeah. Well, I mean, if you look at the virens data and the anolis data, I mean, those are fairly large populations. It doesn't look like they're subject to bottlenecking. I wouldn't say that in all cases. Perhaps there are instances of bottlenecking that can result in these changes.
You compare it to the anolis population in natural that should be much more huge.
Yeah, the thing is, though, even in these disturbed habitats, anolis populations are pretty darn healthy. I mean, there's some other folks working on anolis, and I think they don't seem to be having a demographic problem. I mean, certainly that doesn't go for all species. It's certainly in fragmented, in fragmented habitats. There's often a clear demographic effect where maybe something like that could be going on. So I think there's a lot of questions that we still need to answer about.
But you did not evaluate.
Sorry?
You did not evaluate potential bottleneck signal.
No, haven't yet. No. Yeah.
I think everyone probably in this room would agree that we need to incorporate evolution into conservation planning. But I was wondering, when you use these different modeling approaches, the two you were looking at in the different boxes, do you really get really different results? In some areas you will, but overall is it very, very different? And can you maybe tell us a couple of or maybe one example of where it's like radically different.
Radically different between disturbed and
No, in the conservation planning,
Mhm.
and looking at the areas, you argue for incorporating evolution as opposed to evolutionary processes,
Right.
as opposed to pattern and species richness. But do you really get really different answers in terms of when you actually produce your map?
Yeah, I think we do. I think we're going to. And we're at a very nascent stage.
Mhm.
But, you know, I think overall we're going to find some pretty startling things when we start looking at this. I mean, if you just look at ecotone habitat, which we've been studying for a long time, which we think is very important in generating diversification, you know, about 20% of it is protected. Most of the focus has been on preserving refugial areas where, yeah, there's a lot of species richness in those areas. There's, you know, certainly diversity in those areas, but I don't think that those areas are the engines of diversification in a conservation context, and one that we need to be really concerned about with, you know, maximizing necessarily under a situation of climate change. So I think that I think we are missing a lot of areas. I mean, right here in California, you look at the number of protected areas that encompass gradients in the Sierras. I don't know what the proportion of it is, of those are. There are certainly some, but I think the historical approach has always been to preserve areas that we can define readily, you know.
Yeah, but do the protected areas, since they have all the political constraints when they're designated and practical considerations, do they follow the modeling at all of species richness?
Well, in terms of species richness, oftentimes they do in terms of species richness, sure. In terms of levels of endemism, yeah.
Well, following up on that a little bit, just thinking about California and the possibilities of missing areas that should be preserved. I think about the western zone of Yosemite and of Sequoia Kings Canyon, where the parks ended too early. They didn't get into some of the lower slopes where you've got a lot of species richness and a lot is happening where you get the transitions, you know, the sharp transitions.
Well, like the whole Kern Valley area.
Right.
Yeah.
The Kern Valley area is another one. So there are definitely, you can definitely think of some places. Now, I've just come from a conference where we've been focused on redwoods, and all of the attention is on old growth, which is appropriate right now because so much of it's been lost. But you wonder what else should have been saved in the redwood area. We've gone and saved just the prime old growth.
Right. Right. Absolutely. Okay. Karen?
It's really incredibly neat to hear you're thinking about how to make detailed predictive maps of evolutionary processes, and I noticed you were also looking at pathogens in some of those birds. And from just an ecological perspective, it seems that some of the sharp elevational distributions that were thought to be competitive in birds turned out to be where the avian malaria mosquito could move and where it was limited, which bird was susceptible.
Right.
So do you think a lot of the gradients are going to actually be mediated through unrelated organisms that are interacting in a food web that are constraining birds?
I would think so. I mean, I don't know. Certainly, you know, a lot of diseases, a lot of the vectors are highly sensitive to temperature along these gradients, so I'm sure that, you know, there are many mediated effects that, you know, we're not even touching upon when we look at this. I mean, you know, basically where we are with most of this is, you know, we find these interesting patterns. We don't know what it means in terms of, you know, what the mechanisms are per se. I mean, we can take a hunch with Anolis that it has to do with perch diameter, but in some of these other instances, you know, it's unclear. It's also, you know, we haven't fully integrated demographic information that would be really interesting to know, and looking at haplotype variation of diseases and things like that. Yeah.
Well, sort of following up on that, Tom, from the demographic and population trajectory, and we've attempted to model some of these kinds of things
Mhm.
more mechanistically, and we find ourselves humbled in our ability to predict future for most populations, especially out beyond a relatively short time period,
Right.
20 years maybe. And so I'm wondering how you see bringing in the evolutionary process from that perspective, looking at really long time periods without the ability to make those mechanistic connections. And even if we have them,
Yeah.
we're probably going to be fairly humbled by what
Yeah.
evolution
Yeah.
can produce for us.
Well, I agree. I mean, I agree. It's very much a crapshoot, But I think that what we're hoping is that at least if we can identify areas that have higher evolutionary potential, even if we don't know, what elevation is going to be most important, that we're hedging our bets.
So that's just areas with larger
Higher
variation
turn-
in climate.
I think at this point it's
Over short
areas with
periods.
higher turnover in adaptive traits and genetic traits, and yeah.
Well, in some cases, you can predict the direction it would move given expected climate changes and predictive models. So maybe we ought to be thinking on the leading edge, sort of, as being the critical area to be making sure that you're saving the evolutionary potential. I mean, some things you can't predict which way they're going, but a lot of things you can, and so that would be, to my mind, a first clue as to where starting to look.
You mean geographic edge?
Yeah, like if something's going to shift north, then yeah, you can worry about everything along the whole gradient, but probably the north edge of the gradient is the one that's going to be most critical.
Well, I think, and I haven't read the